So today's topic, packaging of DNA helix. Already the DNA properties and the structure, RNA properties and the structure. The nucleic acid structures and their properties we are discussed. Whether you are taking the DNA helix, that means double stranded DNA. So dsDNA we are taking means. So double stranded DNA we are taking. The two strands are helically they are going to arrange. Helically they are going to arrange, and they are organized or they are arranged within the nucleus. Either so eukaryotic cells you are taking means, so like that as a humans we are taking within the cell. So within the cell, in the nucleus itself, the double stranded DNA material is embedded here or organized or arranged here. So whether you are taking to the length of the double stranded DNA, here DNA length we are taking means, so the DNA length we are taking means, it is considered the distance between, so distance between the two nitrogen bases. So nitrogen bases into number of base pairs here. So that means between the two constitutive nitrogen, that is two successive nitrogen bases, and the total number of the base pairs is giving into the length of the DNA. For example, humans genome we are taking. That means in the humans, the length of the DNA we are taking to the one cell, and their nucleus we are taking. We are having 6.6 into 10 to the power of, so 9 nitrogen bases into the each nucleus we are taking. So each nitrogen base we are taking. So it is having the 0.34 into 10 to the power of minus 9. So nanometers, it is representing as a nanometers here. So nanometers into the distance these are having. It gives approximately 2.2 meters into the length. That means one nucleus, the human cells, their nucleus, the DNA strand we are taking means approximately it is having 2.2 meters here. So like this, each and every organism, their nuclear material, either they are into the eukaryotes means the nucleus, eukaryotes means the nucleic acid is embedded into the nucleus. Whether they are into the prokaryotes means like that we are taking into the bacteria. So for example, E. coli bacteria, E. coli. So E. coli bacteria. Here the specific nucleus is absent, but the genetic material is concentrating into the one certain portion of their cytoplasm here. So much of the length of the nucleic acids, how they are going to organized, how they are going to packed in the particular the small structure of these cells means. So that one only we are going to explain, that is the packing of the DNA helix here. So whether you are taking the E. coli also you are taking means their length of the DNA is 1.34 into 10 to the power of 6 into 0.34 into 10 to the power of minus 9. So an average we are taking means, so this is also the diameter or the length of the E. coli cell and the length of the DNA we are taking, so it is much more number of the times length is having into the nucleic acid in case of the E. coli also. In case of the prokaryotes, like that, the human genome or the human nucleic acid in the cell is having the 2.2 meters of the length. So this much longer nucleic acid, how it fits into the nucleus, how it becomes organized into the nucleus. So that one only giving into the packaging of the DNA helix here. So let us explain into the how this nucleic acid is going to packing. In case of the eukaryotes, we are taking means eukaryotes we are taking means the nucleus. So first D Pro. So the scientist, the D Pro, is explaining the uninema. So structure of uninema. So uninema means single chromatid structure. So that means the length and how the single chromatid is organized into the nucleus, that is explained by the D Pro. And later on Korenberg is explains how chromosomes are organized into the nucleus we are taking. So these two scientists are going to explain how this organization is going to occur. So whether in the case of the eukaryotic organisms we are taking. So already we discussed the human's genome we are taking 2.2 meters of the length it is having. So even though eukaryotes you are taking, the other organisms also we are taking, they are having the specific nuclear membrane. In this specific nucleus structure is there. In this specific nucleus, the nucleic acids are going to arrange here. These lengthy nucleic acids are these lengthy DNA material. So how they are going to organize into in the smaller space or the smaller structure of the cell, that too in the nucleus, means here these are having the packaging, a specific organization is presented, so that only they are embedded, they are going to fit into the smaller nucleus structure into the eukaryotes. Whether you are taking into the prokaryotes. So in the case of the prokaryotes, there is no specific nucleus. Cells are not having into any specific nuclear structure. Just the cytoplasm only presented into the cells. But here the nuclear material we are taking, it is not going to distributed into the entire the cell here. It is also forming as a packing. It is also condensed and arranging as a one specific portion only, either like this, or in prokaryotes the nucleic acid, that means DNA is going to organize or arrange into the circular into the fashion. So this condensed form, it is going to, this is called as a genopore. So this is called as a genopore structure. So like this, without having the specific nucleus, here the nucleus is, suppose we are taking the eukaryotes, here the specific nucleus is there, their membrane is there, so that only it is packed. So we can thought like that. But here we are taking means the prokaryotes, so there is no specific nucleus. But even though there is no nucleus, there is no boundaries of the nucleic acid. The nucleic acid is not going to spreading into the entire the cytoplasm. Why it is? What is the reason behind that? So these are going to organize into in this fashion means. So whether they are going to take, the Korenberg explains why the nucleic acid or the DNA is organized in this fashion, how it is going to organize into in this fashion here. So whether we are taking the double strand DNA material, here a double strand DNA helix we are taking. So these are the two strands are present in the helical structure. So two strands are present. So like this, while the time of the cell division we are taking, the chromosomes are going to forming. So before that, how these chromosomes, how they are going to forming with the help of the DNA strands only. Double helical DNA strands only, they are presented into the chromosomal structures. While into the cell division time, we are going to find out into these chromosomes. So whether we are taking this lengthy double-stranded DNA, how it is going to embedded in a specific small structure of the nucleus in the eukaryotes means. So for that they are having the specific proteins, packing proteins or associated proteins are presented. So with the help of these proteins only, the entire length, that is the lengthy DNA molecule is compactly they are going to organized and embedded in the nucleus here. It is fits for the nucleus. So this is first given. The explanation and the model of DNA packaging is given by the Gorenberg. So what he is going to explain means whether you are taking to the double-stranded DNA. So this double-stranded DNA, they are going to packing by the histo proteins. So basically DNA is negative molecule. So negatively charged molecule here. So it is a negatively charged molecules. So these negatively charged molecules, the histoproteins we are taking, so these are the positive molecules, positively charged particles. The negative charged molecule is attracting towards into the positive charged particles, so that only the double-stranded DNA molecule we are taking means these are going to vipers. So wiping around into so these histoproteins here. So histoproteins, what are the histoproteins? They are involving into the DNA package means two molecules of the histoprotein. Each molecule is contained. So H2A, H2B and H3 and the H4. So these are the four histoproteins forming into the one molecule here. One molecule. So these four subunits together they are forming into the one molecule. So like this two molecules of the histoproteins means, so totally 4 plus 4, 8 octamer. The octamer of histoproteins. So histoproteins we are taking, they are going to participating into the DNA package. For example, here it is into the one histoprotein. Here it is the another histoprotein. Here it is the another histoprotein. So like this, the histoproteins forming as a complex. Around this complex, here it is the one histoproteins. This is the double-stranded DNA. The DNA is going to vaporing around to these histoproteins like this. Vaporing around into these histoproteins. That means it is rounded around the histoproteins because of the affinity between these two charged particles. And when it is going to vapors into around the one histoproteins, how many base pairs are going to vapor around the one histoprotein? Means approximately the 200 to 300 base pairs are going to vaporing around the one histoprotein complex molecule. So like this we are taking means the DNA, the lengthy DNA is going to read around into these histoproteins and forming the condensation. That means their length is going to reducing. So whether you are taking here, so this is the one histoprotein, this is another histoprotein, this is another histoprotein, like this continuously the so many number of the histoproteins. These are all histoprotein complexes. So is going to packing into the lengthy DNA to condensating into a shorter distance. So this way the eukaryotic proteins are, the eukaryotic nucleic acids are going to packing are organized and fits into the nucleus we are taking this. Here between the connectivity we are taking is one histoproteins. This is generally called as a nucleosome. This is the name is given by the Gorenberg. that is called as a nucleosome. This structure is called as a one nucleosome. So between into the two nucleosomes, this is the one nucleosome and this is another nucleosome. So nucleosome, one nucleosome of the DNA is going to connect with the another nucleosome by a DNA fragment, a smaller DNA fragment. So this one is called as a H1. So H1 DNA, H1 fragment is going to linker DNA. It is also called as a linker. So linker DNA here, because it is going to connected between into the two nucleosomes here. So like this, each nucleosome with their surrounding or with another their continuous nucleosome, they are going to connected with the H1 DNA fragments here. So like this, continuously the number of nucleosomes are organized. How they are look like means they are look like as a beads on the string. So whether you are taking into the string, the chain we are going to taking. So in this chain, how the beads are going to organized in the specifically in the certain distance. The same way the string, how it is going to connected with all the beads in a chain. So beads on the string, like that the DNA and the histoproteins are appears here. So finally we are taking, so these are going to fold. How these are going to, so this is into the arrangement, the DNA, double helical DNA structure around into the histoproteins here. And then we are taking this organization is the DNA organization, that is the histoproteins, how many they are going to and how they are going to organize means. So these solenoid, that these nucleosomes is going to fold it approximately 30 folds. So 30 folds are going to form. So 30 folds each turns. These are going to forming as a number of folds are going to form. So these turns are having into the 30 folds. These turns, the 30 folds. So 40 to 50 times. So their length is going to condensating here, because the folds are going to form. So each fold we are taking. So this is into the one fold, this is another fold, this is another fold. So this entire folding is called as a solenoid here. Solenoid. So whether you are taking into the one solenoid, sorry, for example here it is into the one solenoid here. So like this, the 40 to 50 so times the folding is 30 to 40 folds are going to forming, and they are representing into the chromosomal structure. So each solenoid we are taking means how many nucleosomes are presented means 6 nucleosome units are presented. That means here these nucleosomes, these are into the nucleosomes, for example we are taking. So after the 6 nucleosomes, for example this is into the DNA, it is the folding is forming like this. So 6 to 7 nucleosomes are going to forming organized as a one fold here. So like this another fold, so like this another fold. So the entire DNA is forming into these number of solenoids as a solenoid structure. So these are going to fold it, and finally these are going to condensating as a chromosome. So this is the way of the organization, or this is the way of the packing of the DNA helix into the eukaryotic organs. Not only the eukaryotes, even though prokaryotes also. So only the histoproteins are involving in the DNA packaging, means not only the histoproteins, non-histoprotein molecules. So generally these are called as a NHC, non-histoprotein molecule or complexes. They are also associated with these histoproteins, and they are also useful or helpful into the packing of the DNA helix here. So this is a way how the lengthy double stranded DNA helix is going to embedded into the smaller nucleus in case of the eukaryotes, or in case of the prokaryotes, how it is going to condense and organize into the only into the one particular place. It is packing here. So next one, this DNA, how it is going to multiplicate it. So that is going to explain by the DNA replication here. So whether you are taking the cells are going to divide it and giving it the number of cells. So how they are going to multiplicating their number, and each cell we are taking the same number of genetic material they are going to represent it. So how they are going to doubling into their number, their amount of the DNA, how they are going to doubling means. So that is a process is going to explain by the DNA replication. So when we are taking into the S phase of cell division, the S phase of the cell division, so that is called as the synthetic phase. The synthetic phase of the cell division, the DNA is going to replicate it. DNA is going to replicate it here. So to explain into the DNA replication, that is the doubling of the DNA is explaining the doubling of the DNA. There the three hypotheses are going to explain. They are conservative, dispersive, and semi-conservative. So semi-conservative method of replication, conservative method of replication, and dispersive method of replication, semi-conservative method of replication. One by one we can go through. So first one is the conservative method of replication. In this conservative method of the replication, whenever the cells are going to divide it, so for example this is into the one DNA molecule at the time of the cell division. So in this parental DNA, this is into the parental DNA molecule. The parental DNA molecule is unwind first as a two strand. The double helical DNA molecule is going to unwind as a two strands. These two strands are going to synthesized. They are going to synthesize their complementary nitrogen bases. By adding of the complementary nitrogen bases, the new strands, this is the one new strand, this is the another new strand. So new strands are going to synthesizing. Later these new strands are going to combining and forming. As a resultant of the semi-conservative method of replication, after the S phase, the parental strands are forming into the one double helical DNA strand, and the newly synthesized DNA is forming into the another double helical DNA strands. So this is the model called, this type of the replication is called conservative method of replication. That means, so new ones are forming the one DNA, so that is going to the daughter cells, and the two parental strands are going into the one cells. So like this new forms and the parental DNA, so both are going to separately, they are going to enter into the two different cells. This is called as a conservative method. And the dispersive method of the replication we are taking here, in case of the dispersive method, the DNA of the parental strand, so this is the parental strand of the DNA, is going to break down as a number of pieces. So it is going to break down. Later these pieces are going to unwind it and forming. So these are into the parental pieces. So these are going to just here as a dotted lines I am making. So like this we are taking means as a dispersive mode. So dispersive mode, the newly synthesized DNA strands we are taking, so new strands and the parental strands, old strands and the new strands. So combinedly as a dispersive, as a patch like, these are going to organize and forming into the double helical DNA strands are going to forming at the time of the cell division. So this is called as a dispersive mode of replication. And the semi-conservative method of the replication, this is a very important. So this is going to explain by the Watson and the Crick. So presently we are going to following into the semi-conservative method of the replication. So in this, the double-stranded DNA parent, the double-stranded helix of the DNA from the parents is going to unwind first. These are going to unwind and forming as a single strands. Each single strand act as a template. So this is a one single strand and this is into the another single strand. So another single strand of the parents. So this is also parents. So this one is going to acting as a template and this one is also going to acting as a template. So when this one is going to acting as a template, the nucleotides are going to added into the continuously and synthesized into the new DNA strand here. The new DNA template is going to synthesized through the process of the replication. So later on, these two strands are going to helically unwinded and these two strands are going to winded. So that means as a resultant of this type of the replication, the two daughter cells after the cell division is getting into the nucleotide. One nucleotide they are getting, the one is a parental strand, one is the newly synthesized strand. One is into the parental strand, one is into the newly synthesized. So like this is forming into the one cell and this is forming into the another cell. So like this, one cell is going to divide it and forming into the two cells. So each cell is having the one parental strand, one newly synthesized strand, one parental strand, one newly synthesized strand. So like this type of the replication is called conservative method. So both the parent strands are going into the one cell of the daughter, two new strands are going into the another cells of the daughter. So daughter cell, these are the two daughter cells from the one cell of the parent. So when it is going to divide it, so these two cells, one cell is having the completely the parent, one cell is having the completely the newly synthesized. But here, when the cell is going to divide and forming into the two cells, each cell is having into the parent, each cell is having the newly synthesized. So it is called as a semi-conservative method of replication.